Okay, so we are going to continue at this time. We are going to go ahead and complete our third part of the presentation. And just in case we skipped over the screen sharing for part two, here is the images. And we also included the LinkedIn profile of our imaging professional, a deep dive into the field of radiology. And I also included my own LinkedIn profile, which I will be willing to share by the end of this presentation. So as we move forward into part three, we are going to discuss the biological and molecular application of radiology. So modern clinical scientists, researchers use MI to study the processes of how molecular abnormalities found in cells build up to form the basis of a disease. So, this type of study in turn facilitates other important clinical goals. And as we can see here, we have different examples such as the early detection of disease, optimizing therapies that aim for certain molecular targets, predicting and monitoring response to therapy, and also monitoring for disease reoccurrence. And then biotechnology companies also use MI to optimize the drug discovery and validation processes. So, again, MI is molecular imaging, just in case you need a refresher. And we can reference the School of Stanford, which they have a molecular imaging program known as MIPS, which currently houses 26 different laboratories composed of 6 broad sections of molecular imaging and assay development. These sections include chemistry, biology, instrumentation, preclinical, clinical, and nanotechnology. And then within these MIPS laboratories, some research efforts were allocated towards the 5 top areas, including listed here synthesis and validation of radiolabeled and fluorescent molecular probes for molecular imaging, development of molecular imaging instrumentation for living subjects, development of molecular instrumentation, I apologize, molecular imaging approaches, assays for interrogating cellular events in living subjects. So, keynote the inclusion of live subjects for research and the development of software tools for visualization and analysis of MI data. We also have the merger of therapeutics and imaging strategies for improved patient management. So, all to say, an inclusion of each of these key studies, including chemistry, cell biology, instrumentation, post clinical, and nanotechnology, all contribute to the formulation of these ideas listed here. And we also have other things to consider. So, clinical applications of molecular imaging also include the use of nuclear medicine, magnetic resonance imaging, I apologize, resonance imaging, which is MRI, and ultrasound, US. The nuclear medicine applications utilize devices such as a single photon emission, computerized tomography, which is SPECT, and positron emission tomography, PET, or a PET scan. And molecular imaging has 2 basic applications. The first is diagnostic imaging, which is used to determine the location and extent of targeted molecules specific to the disease being assessed. Second is therapy, which is used to treat specific disease targeted molecules. The basic principle of diagnostic imaging application is derived from the ability of cell and molecular biologists to identify specific receptor sites associated with target molecules that characterize the disease process to be studied. So again, the ability of cell and molecular biologists to identify specific receptor sites associated with target molecules. I think that was an important thing to point out, as in my cell and molecular biology classes I've taken this semester of fall 23, all are included. And that just lets me know as a pre-med student that the little things that I'm learning right now can apply later. Every little thing matters, every little concept and things of that nature. And we can move forward at this time. So here I have an image, which I apologize because yes, it is a bit blurry, but I do want to be verbal and point out a few things that we can see. So we see that the imaging goes from decrease to increase dimensions and complexity. We have nanoscopic at the top left. As we approach the right, it goes to microscopic, mesoscopic, and macroscopic. Macroscopic is compared to human medicines, as nanoscopic is the molecular biology of DNA and proteins, and we understand the central dogma of biology, as that is implied here as we read from left to right. And just going across, we have the inclusion of cell biology, pathology, embryology, in vivo, and medicine. Medicine is the final product, or I can say the final product would be just living and the circle of life, but how medicine impacts the quality of lives of people here today. And the level of organism goes from DNA, protein, cell, tissue, embryo, animal, human. And this information regards genomics, proteomics, cytomics, pathology, informatics, cell, and clinical. So here is from a translational research from molecules to humans, which highlights the central role of optical bioimaging. So pharma and biotech are focusing on the DNA and protein level solutions, which is indicated by the green arrow. And the best imaging is at the cellular level, which is at the orange arrow indicated here, which I think that looks pretty cool. It actually reminds me of a galaxy in space, but we can move forward. And the most advanced optical imaging stops at the preclinical level, which is right here in turquoise. And then diagnosis and clinical decision making are based on pathology, while clearly the need and resources peak at the clinical level. So the research aims to superimpose the arrows towards the clinical end while overcoming translational brick walls. And the focus is more so on advanced multi-mode methods to overall help overlapping of these different sections. And expanding quantitative molecular imaging is emphasized for the subcellular microscopy to preclinical studies. So overall, as I emphasized prior, going from such a small scale to a very large scale, every little thing matters as far as the complexity, because it just multiplies in complexity. And I would like to also acknowledge the use of technology, as this imaging was captured from the smaller end of microscopes. And I think that plays a large role in understanding radiology. I can also point out that this can apply to us or research as epigenetic mechanisms such as DNA methylation play important roles of oncogenesis and cancer progression because cancer research is such a heavy research topic. Also, of cellular phenotyping from quantitative DNA methylation imaging to improve cancer diagnostics, prognostics, and even post-operative patient monitoring. Some of these are also used for drug screening. But from here, we can move forward and observe more imaging. So on the right, we have a scale of the change of percentage of post UV exposure. And we have a mouse laying on its back. And this red square really exposes the UV radiation that is present on that mouse. I just wanted everyone to just take a look and see how the scale varies just from a very small spot. Also, we can move forward even further to discuss more into the molecular and biological imaging of radiation therapy and all of those other treatments. And I can also say that the study of the action of ionizing radiation on healthy and diseased tissue is known as radiation biology to seek out nature and just a sequence of events that occur. And some of it is not just to repair or reverse anything that has happened, but really to just better understand what is occurring. And before we move forward with this, I would also just like to speak more on molecular imaging and how it can be assessed for specific molecular targets. And for future reference, specific imaging of such targets can allow earlier detection of diseases and other things that may occur, regardless if it's a situation of childbirth or anything of that nature. So everything is really considered, and we are approaching the end of our presentation. And I would just like to thank you so far for staying here and just learning more about what I'm passionate about. You can go ahead and take out your phone and scan this QR code if you are interested in viewing these radiologic simulations. And I'm just going to go ahead and give a little brief and show what is included on this website. So here it is from the American Society of Radiologic Technologists. And we have these demos here as far as the simulators regarding radiography. We have collimation and centering. We have positioning. We have radiographic anatomy, surface anatomy, and cross-sectional anatomy. So we do not have to play a full video, but I can definitely go through a few seconds of this video to just give a close up of what is being observed. But I highly recommend to take a look and pause the video for yourself to see what is being observed. And I can just say from looking at this image and looking at what is darker, what is brighter, is something that is important to consider. But yes, definitely go ahead to scan this QR code again if you are interested in viewing a little bit more details, what is considered the radiologic simulations. Thank you so much for staying to the end of my presentation. If you have any question regarding the three parts of this podcast, please just take a brief look and screenshot this page. Here are all the links I use for any of the images or any of the key facts and details of what I've discussed so far, again, thank you so much for staying to the end of my presentation, and everyone have a very nice day.